The overall goal of this protocol is to identify host targets that can be manipulated to enhance oncolytic virus therapy.
This method can help answer key questions in the field of oncolytic virus therapy, such as what specific host factors and cellular pathways modulate oncolytic virus replication. The main advantage of this technique is that it allows one to identify host targets that can be manipulated to enhance oncolytic virus therapy in a comprehensive and unbiased manner.
This protocol can also be utilized to identify host factors that modulate virus replication in general.
After developing a high-throughput screening assay according to the text protocol, remove a batch of microtiter plates from the freezer and allow them to defrost for 20 to 30 minutes. In the meantime, fill 2 bottles with 70% ethanol, then fill 2 50 ml conical tubes with PBS and 1 with distilled water. Immerse liquid dispenser tubing into a bottle of 70% ethanol. Then place a piece of masking tape around the tubing to mark the point below which the tubing can be considered sterile. Use approximately 25 milliliters of 70% ethanol to prime the tubing. Pour additional ethanol into the bottle to replace the lost volume. Then let the tubing sit in ethanol for a minimum of 5 minutes. After preparing the cell culture hood, centrifuge the sealed plates in sets at 1026 times G and room temperature for 2 minutes. Next, remove the seals from the plates. Then pipette the required amount of transfection reagent into the conical tubes containing the pre-warmed transfection reagent diluent.